Chapter 9, we are going to discuss the quantification and the analysis of protein. So this is a continuation of the chapter 8. So in chapter 8, we learn how to collect the cell and purify the protein from the cell. And now this chapter, we will learn how to quantify the protein and analyze the protein. So there are many different platforms and methods that we can use to quantitate the protein. However, the spectrophotometry assay of protein in solution do not require either the enzymatic or chemical digestion or separation of the mixture prior to analysis. Just like we use the spectrophotometer method to quantify the amount of DNA. There are three major types of spectrophotometry assays. Number one is UV absorbance method. Number two, dye binding assays using colorimetry and the fluorescence-based detections. Although these spectrophotometry assays can be run at high throughput, they require an appropriate protein standard or constituent amino acid sequence information to make an estimate of concentration. The choice of method used to determine the concentration of a protein or peptide in solution depends on many factors. For practical procedure here that I show you the flowchart, that this flowchart describes the process of selecting the most appropriate assays. Some criteria that need to be considered when selecting an assay including sample volume, the amount of material available to analyze. Typically, fluorescence-based assay provide the best sensitivity and the dynamic range. Microplate assay use lower assay volume but show improved sensitivity, typically up to tenfold when compared with cuvee-based assays. And number two, also some consideration is the sample recovery, right? And so if the sample is limited, a non-disruptive method such as UV spectroscopy may be more appropriate. And number three is the throughput. If multiple samples are to be analyzed, a microplate compatible rapid one-step assay should be considered. Robustness. The absorbance-based dye binding assay appear to display enhanced repeatability and robustness when compared to a fluorescent assays. And also the chemical modifications. Covalent modification, for example, the glycosylation or pegylatation can interfere with a specific assay. And protein aggregation, the solubility of a protein in solution, often a problem for membrane protein or protein prone to aggregation, can alter the expected response for many assays. First one, we will discuss the UV spectroscopy method to determine the protein with absorbance at 280. This method is particularly useful for the detection of protein eluting from chromatography columns because there is no loss of protein. The possibility of nucleic acid contamination should be considered. And remember, when we discuss in chapter 7, so we also use the spectrophotometer to determine the nucleic acid concentration. So for the nucleic acid, the UV absorbance should be at 260, and now this is 280 to determine that the amount of protein in the solution. So the 280 nanometer, basically they can this rely on the amino acid residue with aromatic ring in the protein that they can absorb the light. So this protein, including the tryptophan, tyrosine, these two contribute the most absorbance at this wavelength, and also phenylalanine also contribute to the absorbance at this wavelengths. Advantage of using absorbance at 280 like this method is fast, easily, automatic, and reasonably sensitive. Furthermore, this method does not destroy protein. However, many buffers and other reagents can interfere with A280 spectrophotometry measurement. The concentration limit for those reagents are in this table here. The A280 measurement is also interfered with by the absorbance from the DNA and RNA to the 260, and so sometimes we also need to also measure the ratio between the 260 and 280. So for example, the estimation of protein concentration is valid up to 20% nucleic acid, or an A280 over the A260 ratio less than 0.6. So for a UV spectrophotometer to measure the protein concentration by using absorbance at 280, that would depend the number of the aromatic amino acid in the protein. In the meantime, we can also we can also determine the protein concentration by measurement of the absorbance at the 205 nanometer, and this would be based on the absorbance by the peptide bond. And then this one is more universally applicable than the A absorbance at 280, and the absorptivity for a given protein at 205 nanometer is several fold greater than that at the 280 nanometer. Therefore, lower concentration protein can be quantitated with the absorbance at the 205 method. And the protein concentration can also be determined by the measuring the intrinsic fluorescence based on the fluorescence emission by the aromatic amino acid tryptophan, tyrosine, and the phenylalanine. Usually, the tryptophan fluorescence is measured as the indication of the fluorescence intensity of the protein sample solution. So that would be the detecting protein by intrinsic fluorescence emission. And then the next method would be the colorimetry protein assay technique. So all the colorimetry protein assay technique require protein standard to estimate the concentration of a sample. The ideal protein standard to use in the quantitative assay is the exact same protein in a matched matrix solution that has been assigned using a higher order method, for example like AAA. On the other hand, some commercially available standard may be suitable for use. The most commonly used standard are the bovine serum albumin (BSA) or the bovine gamma globulin, and also immunoglobulin. And of course, in our lab that we will use the BSA as the protein standard. Okay. So the first colorimetry method that I'm going to discuss is the Biuret reaction. The Biuret reaction is based on the complex formation of the cupric ion with the protein. The cupric ion, they form the complex with the protein, as you can see here. In this reaction, the copper sulfate, so you add a copper sulfate to a protein solution in strong alkaline solution, then the purple violet color will be produced, and this is result from the complex formation between the cupric ion and the peptide bond. Color is produced by the formation of a protein copper tartrate complex, like you see here. The Biuret reaction with protein is independent of the composition of protein. Therefore, the protein composition is not a factor. Mainly, it's because the cupric ion interacts with the peptide bond. And then the next method here is called a Lowry method. So as you can see, the first step, of course, the protein, you have copper sulfate, then you have protein, the cupric ion complex, that's the burette reaction. Then you have the second reaction that you add the foreign CO-calcium, and then they will form the protein complex. And the reagent interact, that's because the reagent interact with the cuprous ion and the side chains of the tyrosine, tryptophan, and the cysteine to produce a blue-green color that can be detected between the 650 nanometer and the 750 nanometer. And then another method is called BCA method, benzene-coninic acid assay. And basically, the benzene-coninic acid cuprous complex form a deep blue color that can be read at the 560 nanometer. The BCA method is based on the fact that the sodium salt of a benzene-coninic acid react with the cuprous ion generated by the burette reaction under the alkaline condition. So the first step, so the BCA method is just also like the Lowry and the burette. The first step, they all have to perform the burette reaction. You add the copper sulfate to the protein solution, then the cuprous ion interact with the peptide bond, and then for the BCA, so those the sodium salt will react with the cuprous ion, and then that will give you the deep blue color. Okay, that's a BCA method. Then the breadth method basically is different from the previous three, because the previous three you have to perform the burette reaction, and for the breadth basically it's based on the binding of the Comasi brilliant blue G250 dye to the protein, which result in a dye-protein complex with increased molar absorbance for the determination of the protein concentration. So if you have if it's a more concentrated protein, then you have even the deeper color. That's a breadth acid, depending on the binding of the Comasi brilliant blue G50 dye to the protein. So the more protein you have, the more dye will bind, and then you have a more intense color. For the and then the next method is the fluorescent dye-based assay. So for example, for the fluorescent dye, so we can have this OPA, the phosphate high derivatization reaction. So basically, the rapid OPA, which is the OPA amine reaction in the presence of two capital ethanol, they can produce an intense blue fluorescent product, and that has a maximum excitation and emission wavelengths of 340 and 455. So that's the OPA derivatization reaction. And then the format used in the measurement depend on the throughput, sensitivity, and the precision required of the assay. Traditionally, cuvee have been used for the majority of the spectral. Okay, and Lowry method. So to show you how to generate a standard curve.
Here you can see the test tubes arranged for our standard curve. First, we will adjust the volume of our pipette to 0.2 milliliters. 200 microliters of protein will be added to the first tube. The process will then be repeated, adding twice as much protein to each subsequent tube for our standard curve. Note that our blank tube will not contain any protein. Water will now be added to each tube, including our blank. This will bring all tubes to the same volume, thereby creating a serial dilution of protein concentration.
So this is how you make the standard curve, okay?
We will now add 5 milliliters of alkaline copper reagent to each tube. Each tube will be vortexed gently and then returned to the rack. Will rest at room temperature for 10 minutes.
So that's that. Today.
Next, we will add 600 microliters of the Folin reagent to each tube.
So the first is a Biuret reaction, and now this is second reaction.
Immediately after adding, we will vortex the tubes once more. Tubes will rest for an additional 20 to 45 minutes at room temperature.
Then read the OD.
The blank is transferred to a cuvette for our spectrophotometer and will be used to blank the instrument for subsequent readings. We will then read the absorbance of each tube in our standard curve. Note that we typically read the absorbance for this assay at 550 nanometers and not at 650 as shown in this simulation. In the end, you will have a table like this, which will go in your lab notebook, that has the absorbance values and volumes of each of the reagents. These can be used to generate a standard curve from which you can extrapolate the values for our unknown.
So this is standard curve, so and then you will have a formula, and then from the formula, then you can use it to estimate the unknown protein concentration that you want to determine. So this is the first step. So later on when we perform the experiment, and we always need to set up the protein standard, so that you will set up this standard according to the protocol, and then read the OD, and then generate a graph like this, and then to create the formula. And from the formula, then you will determine or estimate the unknown protein concentration. And then here I will show you the BCA assay.
By measuring the amount of reduced cuprocyan that is generated by chelating copper to proteins in the sample.
So this is the first step, the
reacts
puree,
with BCA,
and then the second
forming
step,
a
the
purple
BCA.
water-soluble complex. The Thermoscientific Pierce BCA protein assay kit includes all reagents needed to accurately determine protein concentration in a sample, including purified bovine serum albumin standard in single-use 1 milliliter ampules for consistent standard curve generation. The kit includes protocols for both microplate and test tube-based assays. However, we will just use microplates in this example. After diluting the BSA standard,
so basically this part our
prepare the BCA working reagent by combining reagents A and B at a 50-to-1 ratio, and pipette 25 microliters of each standard or sample into a microplate well, followed by 200 microliters of the BCA working reagent.
So we don't do the microplate.
Gently mix the components for 30 seconds,
is to show the microplate,
and then incubate the plate
procedure
at 37 degrees for 30 minutes. After cooling the plate at room temperature for 5 minutes, measure the absorbance at or near 562 nanometers with a plate reader. Average all replicate values, and generate a standard curve by plotting the mean standard value.
See, the first step is always to generate a standard curve.
Then use the standard curve to interpolate the concentration of each sample based on its mean absorbance value.
So that's the procedure for BCA assay. And then the next one, the next topic that I want to discuss is the molecular separation through the electrophoresis. So prior to any downstream analysis, purity and integrity are the very first qualities that need to be assessed for any protein sample. This is routinely achieved by the SDS page gel electrophoresis. This technique, associated with a staining method, can detect bands of protein in a sample in relatively rapid manner because it can just finish in just a few hours. So that's the SDS page gel. So for this SDS page gel, basically you have this SDS plus the polyacromide gel, and polyacromide gel that we discussed when we try to analyze nucleic acid. The polyacromide gel can provide this highest resolution for down to the base pair resolution. And then so in the SDS page, so of course we need to have SDS, right? And then to run the protein electrophoresis, and the protein can be separated based on the size. And then, as we know that the whole protein based on the composition of amino acid. It can be somewhere in the some region positive, some region negative. So it's not the unique uniform for the whole protein sample, and therefore the reason we add SDS, you can see the negative charge here, so that we can have SDS to cover the whole protein surface, and therefore we make the protein into the negative charge, and so that will be similar to the nucleic acid. And then we can run the protein under the electric field so they can migrate from negative charge to the positive charge. So that's the purpose that we do the SDS page gel. And then so the SDS page gel, basically it's just like the nucleic acid. You will not be able to visualize the protein, and then until the end of the electrophoresis, then you have to stain the gel so that you'll be able to visualize the protein. So the common method people use is a Comasi brilliant blue. This one is the most frequently employed method for protein detection in SDS page gel. Okay, the Comasi brilliant blue is an anionium triphenyl methyl dye, and so there are two different modifications. One is the Comasi R250 red tint, and the Comasi G50 is green tint. So basically, in the presence of an acidic media, these dyes stick to the amino group of the protein by electrostatic and the hydrophobic interaction. The advantage of the Comasi staining method are the quantitative binding of the dye to protein, and also low price, and also good reproducibility. And for the mass spec analysis, the dye can be removed from the gel with the bicarbonate prior to the tryptic digestion. Therefore, it's very commonly used, and that will be, you can see in the top panel on the right. That's the Comasi brilliant blue stain. And of course, you can use the zinc reverse staining. It's a negative staining, and also you can use a silver staining. Although silver staining has a higher sensitivity, but it's very expensive, so it's not as commonly used as the Comasi brilliant blue. And also you can also use the fluorescence dye to stain the gel. The fluorescence dye gives very wide linear dynamic range over the 4 orders of magnitude. And since the fluorescent staining is an endpoint method, the results are highly reproducible. Several fluorescent dyes such as the Nile Red, IUBPS, Cypro, and Epicococno can be used to reveal nanogram of protein in gel.
SDS page or sodium dodecyl sulfate polyacrylamide gel electrophoresis is an electrophoresis method and very powerful technique in the study of proteins. In SDS page, proteins are separated in a polyacrylamide gel based on their molecular weight.
This is the example of SDS page
For sample
gel.
preparation, a loading buffer containing SDS, beta-mercaptoethanol, bromophenols blue, and glycerol.
So the beta-mercaptoethanol beta-mercaptoethanol is to remove the disulfide bond between the two cysteine, because we are going to break the tertiary structure, protein tertiary structure. We want to make it into the primary structure. Therefore, we have to use the beta-mercaptoethanol to break the disulfide bond. Also, we're going to use the heat to break those hydrogen bond, or ionic interaction, and then also in the presence of SDS will cover the protein so that it will evenly distribute it on the protein surface.
Is added to the protein samples. Protein samples may be biologically derived, for example from prokaryotic or eukaryotic cells, tissues, viruses, environmental samples, or purified proteins. When the sample buffer has been added, the samples are then heated at 95 degrees Celsius for 5 minutes.
So it's 95 degrees to denature the protein.
Proteins
Okay.
are large biomolecules consisting of one or more long chains of amino acid residues. Amino acids are organic compounds that contain amine and carboxyl functional groups, along with a side chain. Proteins are formed by linking amino acids with peptide bonds and fold into one or more specific spatial conformations, driven by a number of interactions such as hydrogen bonds, hydrophobic interaction, disulfide bonds, and ionic bonds. SDS is an anionic surfactant and contain a polar head group with a net negative charge at the end of a long hydrophobic carbon chain. SDS denatures the native proteins by disturbing the hydrogen bonds, hydrophobic and ionic interactions, while the reducing agent beta-mercaptothanol is used to cleave the disulfide bonds. Also, when a protein mixture is heated, SDS binds uniformly to the protein, and the intrinsic charges of this protein become negligible when compared to the negative charges contributed by SDS. This treatment brings the folded proteins down to linear molecules with net negative charge. Therefore, these proteins can be separated in a polyacrylamide gel based on their molecular weight. For gel preparation, a separating gel solution with pH 8.8 and a stacking gel solution with pH 6.8 are used, and both consist of acrylamide, bisacrylamide, ammonium persulfate and T-med. The gel is produced by polymerization between two glass plates anchored vertically in a cassette. The separating gel is poured first. The polymerization reaction is a vinyl addition polymerization initiated by a free radical generating system. Ammonium persulfate is used as the free radical initiator, while T-med accelerates the rate of formation of free radicals from persulfate. These free radicals convert acrylamide monomers to free radicals which react with unactivated monomers to begin the polymerization chain reaction. The elongating polymer chains are randomly cross-linked by bisacrylamide, resulting in a gel with a characteristic porosity. The pore size of the gel is related to the ratio of acrylamide to bisacrylamide. Low concentration is preferred for high molecular weight samples. The stacking gel solution is poured on top of the solid separating gel, and a plastic comb placed on the top of the stacking gel during polymerization enables the formation of small wells in the gel. Once the gel completely polymerizes, the comb is removed, and the gel cassette is placed vertically between two electrodes. Positive electrode located at the bottom of the gel, whereas negative electrode positioned at the top of the gel. The gel is inserted into a chamber, and a tris glycine chloride buffer system with pH 8.3 is poured to allow the conduction of current through the gel. SDS is also present in the gel and in the running buffer to make sure that once the proteins are denatured, they stay that way throughout the run. For sample application, in addition to the samples, a molecular weight size marker is usually loaded onto the gel. This consists of proteins of known sizes, and thereby allows the estimation of the sizes of the proteins in the actual samples, which migrate in parallel in different tracks of the gel. Each sample is pipetted into its own well in the gel, and as we have seen previously, bromophenols blue and glycerol are present in the samples. Bromophenols blue is a dye that is useful for visualizing the sample in the well, and glycerol increases the density of the sample and used to make the sample fall to the bottom of the sample well, rather than just flow out and mix with the running buffer. After the sample application procedure, an electric field is applied across the gel, causing the negatively charged proteins to migrate across the gel away from the negative electrode and towards the positive electrode. Also, small proteins migrate relatively easily through the mesh of the gel, while larger proteins are more likely to be retained, and thereby migrate more slowly through the gel. The stacking gel is used to ensure that all of the proteins arrive at the separating gel at the same time. Because gel wells are around 1 centimeter deep, so in the absence of a stacking gel, proteins would all enter the separating gel at different times, resulting in very smeared bands. As we have seen previously, the running buffer consists of glycine and chloride ions. So, in the sample wells we will have proteins, chloride ions, and glycine. Proteins and chloride ions are negatively charged. Well, glycine can exist in 3 different charge states depending on the pH. The isoelectric point or pHi of glycine is equal 5.97. At this pH, glycine carries neutral charge due to the gain and loss of protons. At a pH below its pHi, glycine carries a net positive charge. And at a pH above its pHi, it becomes negatively charged. The running buffer pH is equal 8.3, which above the pHi of glycine. So, at this pH, glycine is negatively charged. Once the electric current is applied, proteins, chloride ions, and glycine are forced to enter the stacking gel, where the pH is equal 6.8. At this pH, glycine switches predominantly to the zwitterionic neutrally charged state. This loss of charge causes them to move very slowly in the electric field. The chloride ions, on the other hand, move much more quickly. Chloride ions migrate in front of the proteins as leading ions, and glycine molecules migrate behind the proteins as initial trailing ions. Consequently, proteins are trapped and concentrated into a narrow zone between the chloride ions and glycine. This procession carries on until it hits the separating gel, where the pH switches to 8.8. At this pH, the glycine molecules are mostly negatively charged and can migrate much faster than the proteins. Proteins are separated, so higher molecular weight proteins move more slowly through the porous acrylamide gel than lower molecular weight proteins. Due to the relatively small molecule size of bromophenol blue, it migrates faster than proteins. And by optical control of the migrating colored band, the electrophoresis can be stopped before the dye and before the samples have completely migrated through the gel and leave it. At the end of the electrophoretic separation, all proteins are sorted by size and can then be analyzed by other methods, for example protein staining methods and immunological detection, such as the Western blot technique. After removing the glass plates, the stacking gel is discarded and the separating gel can be stained. Chamasi blue is the most popular protein stain. It is an anionic dye, which non-specifically binds to proteins. The structure of Chamasi blue is predominantly non-polar, and it is usually used in methanolic solution acidified with acetic acid. Proteins in the gel are fixed by acetic acid and simultaneously stained. After staining, the proteins are detected as blue bands on a clear background. The molecular weight size markers in a separate lane in the gel can be used to determine the approximate molecular mass of unknown biomolecules by comparing the distance traveled relative to the marker.
So that's the protein gel electrophoresis. In the previous section, we discussed the gel electrophoresis to separate the protein. And now we will discuss the gel-based proteomy, how to separate the protein. Since the SDS page can give a very good resolution peptide, but sometimes a mixture of polypeptides is so complex that we need even better method to resolve them all. And the field that use technique to resolve thousands of polypeptides is called proteomics. Proteomic tools have been exploited to address many molecular biological questions related to various healthy and disease states. Both gel-based proteomics, including the two-dimensional gel electrophoresis, also known as 2DE gel, and the 2DE fluorescence differential imaging gel electrophoresis, DIG, and the gel-free proteomics, such as liquid chromatography and the capillary electrophoresis, are useful methods to identify proteins. And so the following slides, we will discuss the two-dimensional gel electrophoresis and the 2DE fluorescence differential imaging gel electrophoresis, and also the liquid chromatography and the capillary electrophoresis. The gel-based proteomy is the most popular and well-established technique for global protein separation and quantification. Through this technique, overall protein expression of tissue can be analyzed on a large scale, and it's a cheaper approach than the gel-free proteomy. 2D gel MS and the bioinformatics tool are the key component of the gel-based proteomics. The most important step involved in the 2D gel technique are summarized in this diagram here. So basically, we have the biological sample, right, and then we separate through the 2D gel-phoresis, and then after that, we use the software to identify each individual spot, and then after that we can digest the gel and then take it out and then for the further mass spec analysis. And then for the 2D gel, two-dimensional gel, the complex protein sample are separated in 2 dimensions according to their net charge at different pH, which is each protein's isoelectric focusing IEF and their molecular weight through the SDS page. In the first step, the mixture of protein is electrophoresed through a narrow tube gel containing molecule called ampholites that set up a pH gradient from one end of the tube to the other. A negatively charged protein will electrophorese toward the anode. This is because the protein has an overall negative charge after treatment with the heat, SDS, and the reducing agent. As it migrates through a gradient of increasing pH, the protein's overall charge will decrease until the protein reaches the pH region that corresponds to its isoelectric point Pi. Then the protein will stop. And so from this isoelectric focusing, and then we can separate the protein based on their isoelectric point. And after the isoelectric point, then we can do the second electrophoresis. But for the first dimension, also another one is called immobilized pH gradient strip (IPG), which is an integrated part of a polyacromide gel matrix fixed on a plastic strip and copolymerization of a set of non-amphoteric buffer with different chemical properties. And like here in this diagram, you can see the non-linear IPG strip 3 to 5, and then 5 to 7, 7 to 9, or 3 to 7, or 3 to 10, the wide range IPG strip. And also there's another one called a non-equilibrium pH gel-phoresis technique. Can resolve protein with basic to an extremely high PEI from like 7 to 11 that cannot be separated by traditional method. That's the NEPHGE. And then after the first dimension, the second step 2D gel will separate the protein based on their molecular weight using the vertical electrophoretic device with either laminar buffer or trisine buffer. Instead of loading protein sample within the well, the first dimension rehydrated strip is carefully placed on the top of the SDS patch and sealed with agarose. So then the protein can be separated the same. So as you can see from the diagram, so the first dimension we separate the protein based on the isoelectric focusing, and after that we place the strip on top of the SDS patch gel, and then we run the gel, and then the protein from the same PI will be separated based on their molecular weight. And then you can see all the spot on the membrane. So these are all the protein in the sample. And then the next step will be analysis of the 2D image. So you have to rely on the software and the bioinformatics and technique to identify the protein, and also to identify the quantity of the protein, so that you will be able to evaluate whether the protein expression is enhanced or been reduced. And the development of imaging technology has introduced the differential imaging gelatophoresis (DIG) technique. Basically, in the DIG technique, so the sample will be labeled with the fluorescence. So for example, in this case here, we have wild type labeled with Psi3 and the mutant labeled with Psi5, and also we have the internal standard, the poured sample, and then labeled with Psi2 mixed together, then IPG strip, and then after that we have the second dimension, and then we scan the gel at different wavelengths, so we'll be able to see the C2, C5, C3, and then we can compare the expression level directly. The sample buffer for the 2D based on the solubilization, disaggregation, denaturation, and reduction of protein during or after cell destruction is achieved by the use of the chelotropy agents, detergent, reducing agent, buffers, and the carrier ampholytes. For the chelotropy agent in the non-denaturing 2D urea gel, a neutral chelotropy agent such as urea can act as a denaturing agent in sample solution at concentration typically at 8 to 9 mol. It effectively disrupts non-covalent and ionic bond between the amino acid residue. Urea-containing solution may not be heated above 37 degrees Celsius to avoid degradation of urea to a cyanide. Cyanide ion can react with the amide group of protein, which can cause carbamylation and remove the positive charge of the amide, thereby affect the pI of the respective protein. The solubilizing power of urea-containing sample solution can be dramatically increased by the addition of cell urea. Typically, cell urea is used at a 2 molar concentration in conjunction with 5 to 7 molar urea. And then for the detergent, so hydrophobic interaction within the polypeptide chain or between the protein complex can be disrupted in the presence of detergent. The use of detergent can also increase solubility, especially of membrane protein. Non-ionic detergent such as nonylate P40 or tritone S100 can be used. The Rituron ionic detergent CHOPS has much better solubilization, particularly for membrane protein. SDS is excellent in its ability to efficiently and rapidly solubilize protein. And reducing agents cleave the disulfide bond closely within and between the protein subunit, thereby promoting protein unfolding and maintaining protein in their fully reduced state. And the removal of the interfering contaminant. So a sample purity can determine success or failure of any protein analysis. Interfering substances are any contaminant that negatively impact IEF, SDS page, or both. Commonly seen contaminants include salts, detergents, small compounds, nucleic acid, lipids, and polysaccharide. Therefore, it is important to eliminate contaminant from the original biological sample during sample preparation. And then we can use the precipitation method like the trichloroacetic acid, acetone, chloroform methanol, and the ammonia sulfate precipitation. And then ultrafiltration. So protein precipitation with the trichloroacetic acid and acetone, as well as ultrafiltration, result in efficient sample concentration and desalting. And then for the capillary electrophoresis, which is a gel-free proteomic technique. The capillary electrophoresis is a typical gel-free technique with the advantage of superior separation of efficiency, small sample consumption, short analysis time, and automatability. CE can readily be coupled online with MS. CE separate protein with or without prior denaturation. In slab gel or microfluidic channel, protein separation in CE is based on a variety of properties, including their molecular mass, their isoelectric point, or their electrophoretic mobility. In the SDS-CGE, a capillary is filled with gel or viscous solution to form the molecular sieve, and the electroosmotic flow EOF is usually suppressed. EOF is a unique driving force in CE that is generated at the wall of fused silica capillary by the formation of an electrical double layer associated with the ionized silicon group. Therefore, the analytes migrate solely based on their electrophoretic mobility. CGE can be accomplished using either a permanently coated gel or a dynamic coated gel. In the first one, the coated gel, the gel is prepared and bounded inside a fused silica capillary by polymerization of a monomer, whereas in the second approach, the gel is dissolved in aqueous solution to form molecular sieve and used as a separating medium. With the CZE, sample is introduced into a buffer filled capillary either electrokinetically or hydrodynamically. Both ends of capillary and electrodes are then placed into a buffer solution that has also contained the electrode, and a high voltage is equivalent with the in the system. And here is the example of microchip capillary electrophoresis. Basically, in this microchip capillary ME is a miniaturized form of a conventional CE. In the ME, the basic CE configuration has been miniaturized and transferred to a chip format. With ME, it is possible to integrate injection, separation, and detection on a single microchip with typical channel length of 5 cm to 15 cm. So that's the microchip capillary. For the gel-free proteomic technique, liquid chromatography is the fundamental of protein separation science. It can detect molecules at the nanomolar level. Therefore, it is useful in proteomics and the genome research. Many kinds of liquid chromatography, such as reverse phase, high-performance liquid chromatography (HPLC), affinity HPLC, gel permeation HPLC, ligand exchange HPLC, and the capillary HPLC have been used in proteomic research. The principles exploited in HPLC basically are the same as those used in common color chromatography method like we discussed previously in chapter 8. And so here we will just discuss this HPLC by using the same principle. Reverse phase HPLC basically is established on the partition equilibrium between the nonpolar hydrophobic stationary phase and the polar mobile phase, where the polarity of the mobile phase is higher than the stationary phase. And the stationary phase mostly comprise nonpolar alkyl hydrocarbons such as C8 or C18 chain bound to the silica or other inert supports. C18 and the C8 refer to the alkyl chain length of the bonded phase of the column. The chain length influences hydrophobicity of the sorbent phase and thus increases the retention of ligands. C18 is often called the traditional reverse phase matrix because it has the highest degree of hydrophobicity. The reason why C18 is more hydrophobic than other reverse phase is because lengths of the carbon chain are longer. C8 is used when shorter retention time are desired. Lower hydrophobicity means faster retention for nonpolar compound. Therefore, nonpolar compound move down the color more readily with C8 than with C18. Affinity HPLC is a chromatography method capable of separating biochemical mixtures of highly specific nature, so that we can design a stationary phase that reversibly binds to a non-substrate molecule just by combining affinity chromatography, just like we discussed previously. So you can have this antibody protein binding, and then you can reversibly to remove the protein from the column. So that's affinity HPLC. For the gel permeation HPLC, it's just like the size exclusion chromatography. So basically, the principle the gel permeation HPLC work on the principle of size of compounds. So it's exactly the same as size exclusion. So in this technique, the large molecule are eluted first, followed by smaller molecule. The stationary phase is a porous solid such as glass or silica, or a cross-linked gel that contains pores of appropriate dimension to affect the separation desired. So the large molecule will be excluded, so that will elute first, and then smaller molecule will be the last. And the ligand exchange HPLC is an advanced version of reverse phase HPLC, where the reverse phase column is replaced by an ion exchange column. So the ion exchange, in the ligand exchange HPLC, either the anion or cation exchange column is used, but mixed columns are also available that improve the separation efficiency. In the cation exchange chromatography, the stationary phase is usually composed of the resin containing a sulfonic acid group or carboxylic acid group of negative charge, and therefore cationic molecules are attracted to the stationary phase by electrostatic interaction. In an ion exchange chromatography, the stationary phase is a resin generally containing primary or quaternary amine functional group of positive charge, and therefore this stationary phase group attract the solute of the negative charge. So these are the ligand exchange HPLC. For the capillary electrochromatography, which is a hybrid technique of HPLC and the capillary electrophoresis, and this is the capillary electrochromatography, combine the high peak efficiency that is characteristic of electrically driven separation with high separation selectivity. And here is just the summary of the various chromatography techniques for proteomic analysis. So on the left side, you can see the reverse phase HPLC, and then in the bottom here, affinity HPLC. And then right side we have ligand exchange, high performance liquid chromatography, and then also we have the CEC here. So that will be the separation of the protein. And then after the protein are separated and quantified, the next step is to identify each protein in the sample. So for example, individual spots are cut out of the gel and cleaved into the peptide with proteolytic enzyme, and this peptide can then be identified as mass spec. And mass spec exploit the difference in the mass to charge ratio of ionized atom or molecule to separate them from each other. The basic operation of mass spec is to, number one, evaporate an ionized molecule in a vacuum, creating gas phase ion, and number two, separate the ion in space and time based on their M to Z, which is the mass to charge ratio. And then number three, measure the quantity of ion with specific M to Z ratio. Different mass spec have been developed depending on the method to ionize protein for analysis. The two most prominent mass spec methods for protein analysis are electrospray ionization MS, ESI MS, and the matrix-assisted laser dissociation ionization time-of-flight MS. It's called Marty-Topf MS. So now we will talk about these two, the protein analysis method. And the first one is the ESI MS. A solution of macromolecule is sprayed in the form of a fine droplet from a glass capillary under the influence of a strong electrical field, and the droplet pick up positive charge as they exit the capillary. Evaporation of the solvent leaves multiplied charge molecules. Individual protein molecule acquire about 1 positive charge per kilodalton. The mass spectrum of this charged molecule is a series of sharp peaks whose consecutive mass to charge value differ by the charge and mass of a single proton, leading to a spectrum of the mass to charge ratio for a single protein species. Computer analysis can convert this data into a single spectrum that has a peak at the correct protein mass. ESI-MS recently is evolved to the so-called nanospray ionization, while the tandem MS is rapidly replaced by cascade of mass spectrometer using more sophisticated and sensitive detector and accommodating other intermediate sequencing instrument like electron transfer dissociation. So this is ESI-MS. And then the Marty-Topf. In Marty-Topf method, a peptide is replaced on a matrix, which causes the peptide to form the crystal. A laser pulse is used to excite the chemical matrix, creating a microplasma that transfers the energy to protein molecule in the sample, ionizing them and injecting them into the glass phase. An increase in voltage at the matrix is used to shoot the ion toward a detector. Protein molecules that have picked up a single proton can be selected by the MS for mass analysis. This is because the time it takes an ion to reach the detector depends on its mass. The higher the mass, the longer the time of flight of the ion. In a Marty-Topf MS, the ion can also be deflected with an electrostatic reflector that also focuses the ion beam. Therefore, one can determine the mass of the ion reaching the second detector with high precision, and these masses can reveal the exact chemical composition of the peptide. As such, the Marty-Topf MS is very sensitive and accurate for determining amino acid sequence. So that will be the end of the chapter. Chapter 9.
MALDI-TOF mass spectrometry is a versatile analytical technique to detect and characterize mixtures of organic molecules, especially large synthetic polymers and biopolymers. MALDI stands for Matrix-Assisted Laser Desorption Ionization. This ionization method involves a laser striking a matrix of small organic molecules to make the large, fragile analyte molecules into gaseous ions without fragmenting or decomposing them. TOF stands for time of flight, a mass spectrometry method that separates ions by their mass-to-charge ratio and determines that mass-to-charge ratio by the time it takes for the ions to reach a detector. Let's look at each of these components of MALDI-TOF spectrometry in detail. Like any form of mass spectrometry, the MALDI-TOF process first involves ionization of the analyte molecule. The analyte is the molecule or mixture that's being analyzed. MALDI-TOF is best suited to analyzing biomolecules like peptides, lipids, nucleotides, saccharides, or other organic macromolecules. Because they're so large and can decompose when heated, traditional ionization techniques like electron impact will fragment or destroy macromolecules. MALDI is considered a soft ionizing technique that ionizes analyte molecules whole. Key to the MALDI ionization process is the use of a matrix. The matrix is a small organic molecule used to facilitate the ionization process. A few common examples are shown here, such as CHCA, DHB, and SA. Common to all matrix molecules is a conjugated pi system, like a benzene ring and attached functional groups. This conjugated pi system is able to absorb energy in the form of UV light and blast the analyte molecules into the gas phase. Solutions of the matrix and analyte are deposited onto a sample plate known as the target. The target can be made of polished or ground stainless steel and has spots for several different samples to be applied. The solvent is allowed to evaporate from the sample solution, leaving behind a solid sample spot on the target that will be analyzed by the MALDI-TOF spectrometer. Once inside the spectrometer, the target is brought into a vacuum and struck by a pulsed UV laser, usually at a wavelength of 337 nanometers. The laser energy is absorbed by the matrix molecules, which are energetically ablated from the surface of the sample, carrying the analyte molecules into the gas phase as well. During the ablation process, the analyte molecules are ionized, usually by proton transfer with the nearby matrix molecules. The most common MALDI ionization format is for analyte molecules to carry a single positive charge. Now that the gas phase analyte ions have been generated, their masses can be analyzed by the time of flight mass spectrometer. The time of flight process accelerates the gas phase ions in a high voltage electric field. This field imparts a constant amount of kinetic energy on the ions. Because kinetic energy is equal to 1/2 mass velocity squared, a constant kinetic energy will cause the smallest of the ions to travel the fastest. In this example, we have a mixture of 3 macromolecules with masses of 1, 2, and 4 kilodaltons, where 1 kilodalton equals 1,000 atomic mass units. Using standard TOF conditions, this would cause the smaller 1 kilodalton ion to be accelerated to the fastest velocity, around 54 kilometers per second. The 2 kilodalton ion would be accelerated to around 38 kilometers per second, and the largest 4 kilodalton ion would be accelerated to the slowest velocity, around 27 kilometers per second. The TOF detector then records the time it takes for the groups of ions to travel a certain distance. From our previous examples, for a standard 1.5 meter flight, the smallest ion would reach the detector in around 28 microseconds, the next at around 39 microseconds, and the largest ion in around 56 microseconds. From these times, and the known constant kinetic energy of the electric field, The spectrometer is able to calculate and record the mass-to-charge ratio of each ion it detects. MALDI offers a number of advantages over other forms of ionization, such as electron impact. The most important advantage is the fact that large organic molecules, such as synthetic and biopolymers, simply cannot be brought into the gas phase and ionized by conventional gas chromatography. The relatively recent introduction of MALDI and electrospray ionization has allowed for the study of molecules that were previously too large, polar, or otherwise non-volatile to be ionized in the gas phase. MALDI is considered a soft ionization method, meaning the complete molecular ions remain intact when ionized. Harsher ionization, like electron impact, can cause the molecules to fragment. This soft ionization makes the direct mass spectrometry of mixed samples possible. When only the molecular ions are observed, the mass spectrum shows the distribution of the different particles in the sample arranged by their mass-to-charge ratio. For mixed samples with fragmented ions, the overlapping fragment masses would make identification of the individual components very difficult. Because there's no heated injection chamber, like in other forms of ionization, MALDI allows for the analysis of sensitive molecules that would decompose under high temperatures. Finally, MALDI allows for the preparation and storage of a solid sample. Because the solvent is allowed to evaporate from the matrix analyte sample solution, a solid sample spot is left behind on the target plate. MALDI offers versatile molecular analysis with broad-ranging applications.